My name is Dr. Andrew Lloyd. I'm a UKRI Future Leaders Fellow at Aberystwyth University, and I'm going to talk to you today about some of the research that we do in my research group, and also how our research fits into some of the wider research that goes on at Aberystwyth University. So the theme for this year, for 2022, British Science Week, is growth, which is quite fitting for a lot of the research that we do around plants and how they grow. In my particular research group, we are interested in a specialised cell division that happens in the reproductive structures of plants. It's a cell division called Meiosis that gives rise to pollen and egg cells, and so I'll talk a little bit more about that later. But first I'll give you a little bit of an overview of our institute. So this is IBERS, just outside Aberystwyth in mid-Wales. At this research institute there's a lot of plant breeding and plant science research that goes on. And Aberystwyth has over 100 years of history of plant breeding. And that breeding focuses on a number of different crops. It includes oats, are a big one. Also quite a few forages, so we have ryegrass and clover. Well, you can see in this photo here, all the green hills of West Wales, there's a lot of ryegrass and clover growing around here. And also, more recently, work on the bioenergy crop miscanthus. And so breeders at the university spend quite a lot of their time generating new potential varieties, but before they can go out to market, they need to be tested to see which potential varieties have the specific traits that we need. And so a lot of work gets put into assessing the traits of those plants, or the phenotype of those plants, and so we call this phenotyping. And that happens at a number of different scales. So in our research group we look quite often at single cells and look at what's going on even at the subcellular level. But there are researchers at the Institute looking at individual plant structures. So this is a micro CT scan of a wheat spike. We also have the National Plant Phenomics Centre at Aberystwyth University, and there we have a number of smart glasshouses where we can run plants through and subject them to different treatments and see how they respond. And so we have a small system here where we can run smaller plants or seedlings through, but we also have a larger system that we can run whole plants through, like oats or wheat or barley or things like that. And then of course we can grow these plants outside in the field and see how they respond in real world conditions. So we have a little bit of a flyover of the Institute. Down there below, straight away you can see one of the experimental field trial areas. And then we come and fly over the research buildings, and we're sort of nestled here in between the mountains and the sea towards the west of Wales. If we pop down now inside one of these buildings, we can enter into the smart glass houses, and so you can see here an array of brassica plants that are growing inside this facility. And we can hop on board one of these plants. So each plant is on its own individual, essentially train car that gets carried around on a conveyor belt system, and it automatically gets taken and gets watered, but also it gets taken through this series of imaging chambers. And there are a number of different imaging setups that use different types of cameras to photograph these plants. And as you can imagine, when you've got a large population of plants that are growing for several months inside this facility, it generates a huge amount of image data. And so there are a number of computer scientists and data scientists that work within the Phenomics Centre to analyse that image data and extract interesting information from those images, like how plants are growing and how they're responding to different treatments that we might want to subject them to. If we pop back outside again, then we can see one of the field trials. So this is an oat field trial that we're flying over here. And using drone imagery, researchers are able to recreate 3D models of some of these field trials, and then we can extract useful information about how the plants are growing from these 3D models as well. But if we take a bit of a step back for a minute, humans have been interacting with plants and growing and cultivating plants for an extraordinarily long time. And as soon as humans started selecting plants that grew best, or particular plants that had the characteristics they want, indirectly they've been altering the genomes of these plants. And so in a sense, plant breeding has been going on for over 10,000 years. So on here we can see this is a spike of common wheat. So this is our standard wheat that gets grown. And you can see it has a nice thick head where there's some nice big plump seeds inside there. On the left are the three different wild species from which wheat evolved. And so you can see these are much thinner, much more grass-like structures with much tinier seeds. Another big, innovation that came along with domestication of these crops was preventing the seed head shattering. So if you're a plant growing out in the wild, then you want to shatter your seeds and spread them as far and as wide as you can so that you can get your offspring growing in new places. But if you're wanting to collect that seed, to harvest that to eat, then ideally you want all of that seed to remain intact in one spike sitting on the top of a wheat plant so you can come along and easily harvest it. So that was one innovation. There are other innovations to do with the structure of the wheat spike itself, which made it easier to extract the grain from the rest of that plant material. Another thing that's changed a lot in particularly the last 100 years of a more scientific approach to plant breeding is plant height. So this is an example from wheat, and we can see varieties that were released from 1910 through to 2010. And you can see that during that time, wheat varieties have had a huge reduction in size. And so the vast majority of wheat that is now grown are these dwarf varieties. And there's a number of reasons for this, but the shorter plants tend to be a bit stockier and stouter in stature, and so they don't get blown over as much by the wind, and so we call that lodging. And because of that, we can get better yields for them, particularly in windy environments. And also because they're putting less energy into growing really big stems and really big leaves, then there's more energy devoted to putting into those grain, and so we can get bigger yields as well. So plant breeding has continued to improve our crops and improve the amount of crops that we can grow. If you look at this graph here, this is showing us the amount of grain that can be produced from a set area of land. And from the 60s through to the mid-90s, we saw there was continual increases in the amount of yield that we're able to, or the amount of food that we can produce from the same area of land. And there were a number of reasons, but one of the big ones is plant breeding, which has improved the yield that we're able to achieve. But since the mid-90s, these yield gains have slowed down, and you can see that essentially plateaued, and in some cases possibly even fallen away. And one of the big reasons for this is a change in climate. And so plants that were well adapted for conditions of 20 years ago are not so well adapted for the conditions of today. And so we have increasing global temperatures, we also have increasing variability in our weather. Along with these changes in the environment comes changes in disease pressure, so we have new diseases that are coming in all the time, for which our crops are not particularly well prepared. And at the same time, if we want to grow enough food sustainably, we also want to be thinking about the wider ecosystem. And for that reason, we want to be limiting the amount of chemical inputs that are going into the fields. So we want to be reducing things like pesticides and herbicides. So all of this presents challenges for growing crops. And one of the big problems with crops is that they lack diversity. So if you look at a wild population of wheat, for this example, individuals are really variable. And there are a lot of traits that vary between each individual. And because of this diversity and this variability, within that population, there's resistance to a wide array of different diseases and lots of different stresses that population might encounter. And also because of that diversity, it makes these populations quite adaptable, because there's a lot of potential traits out there that can be drawn on in natural selection. In contrast, our cultivated crops are more or less clones. And on the one hand, this is fantastic because it means all of those plants come to peak ripeness at exactly the same time. All of the seed heads are at exactly the same height, so it's easy to harvest with our harvester. All of the grain is always of the same quality year in, year out, which means bakers know exactly how their dough is going to respond when it goes into the oven. But there are downsides as well, and one of those downsides is that because they lack diversity, they have resistance to a smaller set of diseases, and they're also able to cope with a smaller array of different environmental stresses. This lack of diversity means that they're essentially set in stone and they are unable to adapt to new conditions. And so part of the role of plant breeding is to introduce new variation back into our cultivated varieties. So there are some new ways that are potentially coming online that are just beginning to be looked at in terms of how we could add new variation. And one of these is looking at gene editing through new technologies like CRISPR-Cas9. But today I'd like to talk to you about a more traditional plant breeding approach, which is to go back to these wider populations and see if we can reintroduce some of the traits that are already out there in these wild populations. So this is a map showing us where the biodiversity of wild wheats is located. And you can see by this red mark here that the real biodiverse area is in the Middle East. And so as you can imagine, there's a lot of wild wheats out there that are well adapted to growing in hot and dry conditions. There are also, as I mentioned earlier, a lot of disease resistance traits in these wild populations. So this is an example showing, so here on the left is a leaf from a wild wheat, and it's been inoculated with a certain pathogen, but this wild wheat is resistant to that pathogen. In contrast, the next three leaves are of a cultivated wheat variety, and you can see that they're definitely not resistant, and so you can see the disease spreading all over these leaves. Then the last three leaves are cultivated wheats, so things that would be grown commercially, but the resistance trait from the wild wheat has been added into these cultivated varieties. And so now this cultivated variety is resistant to this particular pathogen. So this is the approach that we'd like to be able to use, and as a result we can see that these wild relatives of crops and also something we term land races are really important because they're a fantastic resource of potential new traits that we could add to our breeding. And so because of that, for decades now, researchers across the globe have been really looking to safeguard the diversity of the world's crops and crop relatives. And so one of the places that these seeds are stored is in the Svalbard seed vault. So this is up in the Arctic Circle, and it's a seed vault that holds millions and millions of samples of different seeds that have been collected from populations all around the world. And it includes cultivated varieties, but it also includes all of the relatives of the crops that we know of. And so this is a really fantastic resource that breeders or scientists can go back to and request seeds from. So we don't have anything quite that impressive in Aberystwyth, but we do have a fantastic seed store as part of our new Aber Innovation Campus. And so we have, as part of this new building here, we've reinvigorated our seed store, and we have now over 30,000 seed samples of seeds that have been collected over the course of more than 100 years from all around the world, and also that have come out of breeding programs run at Aberystwyth for over 100 years. And so there's a fantastic resource here, particularly focused on the crops and relatives of crops that we work with in the Institute. So what then is the next step after you've identified potentially a wild parent that has a new trait that you'd like to introduce into your crop? Well, the first step is to cross these two different plants together. And so to do that, we take some pollen from one parent and use that to fertilize the flowers of another parent. And so you can see a very old image here of some crossing of ryegrass that's going on in the Institute, but it happens essentially exactly the same way today. So each of our parents, like most organisms, has two copies of each of its chromosomes. So parent 1 has two copies of each chromosome, and parent 2 has two copies of each chromosome. And these chromosomes are the big structures into which all our DNA is packaged inside the cell. And so when we generate the next generation from these two parents, the offspring, then parent 1 will donate one copy of each of its chromosomes to the hybrid, and parent 2 will donate one copy of each of its chromosomes to the hybrid. And so we refer to the offspring of these two individuals as an F1 hybrid. And so the F1 hybrid displays quite often an intermediate set of traits between a sort of mix of the traits in parent 1 and parent 2. But importantly, what happens in the F1 hybrid, in the reproductive structures, in the flowers, is a process called meiotic recombination. And so in meiotic recombination, segments of the chromosomes from parent 1 and parent 2 get swapped around inside the F1 hybrid to generate a new chromosome. We've got that over here on the right. And this new chromosome has some sequence that came from parent 1 and some sequences that came, parent 2 and some sequences that came from parent 1. And so this is an entirely new chromosome that has a new set of traits on it. And so it's this process of meiotic recombination that really generates the new combinations of traits that we see in our plants. So in all sexually reproducing organisms, there's two main types of cell division, mitosis and meiosis. So mitosis is the one that's important for vegetative growth, and that's going to grow us new roots and shoots and leaves. And in that, each cell undergoes one round of DNA replication and then one round of cell division. And so at the end of mitosis, you end up with two cells, and each of those two cells are an exact copy of the cell that went into mitosis. But the cell division I spoke about just before, meiosis, is this specialized cell division that only occurs inside the reproductive structures and gives rise to pollen and egg cells. And so in meiosis, we start with a cell that undergoes, again, one round of DNA replication, but this time it has two rounds of cell division. So we have the first round of cell division where we separate homologous chromosomes into the different daughter cells, and then the second round of cell division where the sister chromatids of each of those chromosomes get separated into two daughter cells. So at the end of meiosis, we now have 4 cells, and each of those cells is genetically unique, so it has a sequence that has never been seen before, and it only has one copy of each chromosome, so it has half the amount of DNA that was present in the original cell that went into meiosis. And the reason all of these cells are unique is because of this process of recombination that takes place, where segments of chromosomes get swapped. And that process of recombination occurs quite early on in meiosis 1. We study meiosis and meiotic recombination in our lab, and so to do that we spend quite a lot of time at the microscope staring down at cells that are undergoing meiosis, and we can see some cells here. So these are in white, you can see the DNA, and at the beginning of meiosis, when the chromosomes are just beginning to condense, we can see these thin strands of that represent the individual chromosomes. And at the beginning it looks just like a big jumble of spaghetti all mixed together, but as meiosis progresses, homologous chromosomes find each other and pair up along their entire length. And then those pairs of chromosomes begin to condense and get more and more tightly packed until we form these structures that we call bivalents here. This is coming into metaphase 1 in meiosis. And so this particular plant that we're looking at here has 13 pairs of chromosomes. Each of these white shapes represents one pair of chromosomes. And then in anaphase, in the next step of meiosis, these pairs of chromosomes separate out to the two different daughter cells. And then, so that's the first meiotic division, and we undergo the second meiotic division where the sister chromatids separate to new daughter cells. And so at the end of meiosis, we now have 4 cells. And so now each of these cells will go on to produce, in this case, a new pollen grain. We can also track the location of specific proteins of interest in these cells. So this is, again, a cell going through meiosis, and we can we're looking at 2 different proteins here. One marked in green is one of the first proteins that loads onto the chromosomes at the beginning of meiosis, and you can see again these long thin lines of all mixed together of the different chromosomes. But then there's a second protein that we've marked in pink here, which is found where pairs of homologous chromosomes are pairing up along their length. So you can see as meiosis 1 progresses, we get more and more of the chromosomes are becoming completely paired along their entire length. So we can build a sort of 3D reconstruction of one of these cells. And so in blue, here's the DNA, and we take that away, and then we can see more clearly now the individual chromosomes that are joined in pink. And then there's one pair of chromosomes that still hasn't completely joined yet, so we still see some green there. Okay, so we've got our parent 1, we've got our parent 2, we've got pollen from one to fertilise the other to make our F1 hybrid. And in that one F1 hybrid, there's recombination taking place, which is swapping segments of chromosomes to generate entirely new chromosomes that have new combinations of sequence. And depending on which segments of chromosomes come from parent 1 and which segments of the chromosome come from parent 2, this will mean we get different combinations of traits in the offspring of this F1 hybrid. So what I'm showing you here is just a representation of what's happening for one chromosome, but of course this is happening for every chromosome in the genome of that plant. And so from the progeny of the hybrid here, then we can generate families that are potential new varieties. And so the next step then is to grow these potential new varieties out in the field and assess how they respond and how well they grow and what the quality of their grain is. And so if we pop back outside again now, we can see a field of oats that is now, it's time to harvest, so all the seed is ready. And you can see the individual rectangular blocks where these different potential new varieties are being grown. And so this small little harvester is designed so that it can harvest and keep the seed from each of these separate experimental plots. And then once all that seed has been collected, then it will be taken back, and the breeders will assess how much grain each of these plots produced, what is the quality of the grain, and decide which of these potential new varieties will then be taken on to the next step and be bulked up so that we've got enough seed to send out to market and for it to be grown by farmers across the UK. So that sort of brings us more or less to the end of my talk. So I'll leave you with a couple of take-home messages. So the first is that for sustainable agriculture going forward, we need to breed new varieties that are adapted not to conditions now, but to the future conditions that we might be meeting in 10 or so years' time. And so we need to be able to ensure high yield, quality and resilience of our crops in a time when we're having environmental change and needing to reduce our chemical inputs. So to breed new varieties, that is done by assembling combinations of traits from 2 or more parents. And so these could be 2 different varieties, or land races, or wild crop relatives. And this diversity of crops and crop relatives is a really important source of new potential traits. And at the cellular level, it's meiotic recombination that combines DNA sequences from 2 different parents to generate new and unique chromosomes that have new combinations of traits. And with that, I would like to say thank you for listening. If you're interested in any more of my research or that of the wider institute, then do follow the website there. Well, goodbye, thank you for listening, and I hope you had a good time.